Okay, welcome back. So this is part 2 in the muscular system video. In part 1, we set some background information. So we talked about different parts of a muscle and the levels of muscle complexity. We looked at a sarcomere, and we looked specifically at what was going on in terms of actin and myosin and our sliding filament model. So now in this video, we're going to put it all together, and we're going to talk about those things that have to be involved with getting a muscle to actually contract. Now, please pay attention, because when we talk about the major steps in the contraction of a skeletal muscle, there are 2 important steps. The first of which is stimulation of a muscle fiber by a motor neuron. So before a muscle fiber can actually contract, a motor neuron actually has to stimulate it with an action potential. After it's been stimulated, then the muscle can contract using what we call excitation-contraction coupling, using what we learned in the sliding filament model, where those actin and myosin slide over one another. Now, please be aware that each one of these 2 main steps, stimulation of a muscle fiber by a motor neuron and excitation-contraction coupling, each one of these is a potential essay question on the next test. I can tell you from experience, students tend to want to smush these 2 things together. But I am going to be very deliberate in going through these as separate steps. Please be aware as I'm doing this, and for goodness sakes, if you want to do well on this next lecture test, you'll want to be practicing each one of these steps individually, because there will be 100% guarantee that there will be either an essay question or multiple choice and fill in the blank questions on this stuff. So it is good stuff to know. Okay, so let's start in the beginning with stimulation of a muscle fiber by a motor neuron. How could this essay question be written? Well, typically it would be something like identify and explain the major steps in the stimulation of a muscle by a motor neuron, including the parts of the neuromuscular junction, which we're talking about in a moment, and a description, definition of those parts and how they work. For excitation-contraction coupling using the sliding filament model, it might be written like this. Identify and explain the steps in excitation-contraction coupling using the sliding filament model. Okay? And it's the purpose of this video to go over that information with you, so you should be using this in conjunction with the PowerPoint that has been put in Canvas. All right, so step number 1, stimulation of a muscle fiber by a motor neuron. So the muscle fiber has to be stimulated to contract by an action potential from a motor neuron. And do we remember what an action potential is? A wave of depolarization and repolarization, right? All right. Now, interestingly enough, where the motor neuron comes close to the muscle fiber, we've got an arrangement called the neuromuscular junction. And we call it this because the motor neuron doesn't actually physically touch the muscle fiber. There's actually a little gap called the synaptic cleft in between them. As a result of that, since we know an action potential is a wave of depolarization and repolarization traveling down our membrane, if we've got a gap in between these 2 cell membranes, then we're going to have to do something to allow the message to go from the motor neuron delivering the stimulus to the muscle fiber that is receiving the stimulus, which will then contract. So it's important to know about how this neuromuscular junction functions in order to understand what's going on here. And as you can see from the picture here, here's our motor neuron. And we'll talk about why it's wrapped in a little membrane right here coming up in a little while in a future video. And then here we see the terminal end of that motor neuron stimulating different parts of our muscle fiber. And here's an enlargement of that area right here. So we see here's our motor neuron in kind of the greenish color. Here's our muscle fiber in kind of the pinky orange color. And then we've got this gap in between them, which is our neuromuscular. Well, the gap is called the synaptic cleft. And this entire association here is called the neuromuscular junction. Neuro for neuron, muscular for muscle. So the first part of our neuromuscular junction is called the synaptic terminal. And the synaptic terminal is kind of this bulb-shaped distal end of our motor neuron. And stored within the synaptic terminal are going to be chemicals that are generally called neurotransmitters. The particular neurotransmitter that we're going to find in the neuromuscular junction is called acetylcholine. And yes, that is something that you do need to know how to spell. And the proper abbreviation for acetylcholine is capital A, capital C, lowercase h. The proper abbreviation for acetylcholine. And it even says, write out the word acetylcholine and the abbreviation. So when we're using abbreviations, okay, you always have to write out the word first and put the abbreviation in parentheses afterwards, and after that you can use the abbreviation within a particular question, right? So don't make the mistake. I had a student do this one time that there was a fill-in-the-blank question that the answer was acetylcholine, and they did that. But then there was an essay question that also involved acetylcholine, and the student did not write the word out with the abbreviation. And the student's rationale was, well, I already did it. Unfortunately, the way I grade things, I don't always go right from the fill-in-the-blanks to the essays. So for every different question, you need to write out the word and the abbreviation appropriately. And as I mentioned, the gap in between the motor neuron and the muscle fiber is called the synaptic cleft, and it is literally just a space. And then the last part of our neuromuscular junction is called the motor end plate. And some resources use a slightly different term. I prefer that you use the term motor end plate, which is the part of the muscle fiber that's going to have receptors for the chemical acetylcholine. And what are those receptors? They are chemically gated sodium ion channels. Now I know we got to think back to when we talked about action potentials, right? We said in order for an action potential to occur, you have to open up gated ion channels and gated sodium ion channels and allowing sodium to rush into the cell, making the inside of the cell positively charged. That's depolarization. Well, in the neuromuscular junction, the motor end plate, these have chemically gated sodium ion channels. So when acetylcholine is going to be released, it's going to diffuse across the synaptic cleft, plug into those receptors, and those receptors are chemically gated sodium ion channels. So when acetylcholine is going to plug into them, they're going to open, sodium rushes into the cell, bada bing, bada boom. We have depolarized that one part of the membrane, our motor end plate. So maybe you're beginning to see how this mechanism is going to work to allow the action potential to go from the motor neuron to the muscle fiber. And here is a rather crude drawing that I made, but it's something that you should be able to do yourself. Being able to draw and explain the parts of the neuromuscular junction are always a good idea. So here we see this bulb-shaped part of our motor neuron, which is the synaptic terminal of our motor neuron. We've got our gap or our space in between these two cells, and that is called our synaptic cleft. And here we see a little depression in our muscle fiber in which we would normally find these chemically gated sodium ion channels, our acetylcholine receptors. And we call this area the motor end plate of our muscle fiber. So be able to draw something like this if asked. And here again, we saw part of this picture before. So here's the motor neuron stimulating our muscle fiber. Here we're going to blow up this area right here so you can see the synaptic terminal clearly. You can see the synaptic cleft, and you can see our motor end plate. And now they're putting this box around this here so we can see it even more closely. We've got our synaptic terminal here with these vesicles that are full of acetylcholine. We've got our synaptic cleft, and on our motor end plate here you can see very clearly our receptors. So when we're talking about stimulation of a muscle fiber by a motor neuron, We said this is a possible essay question. I don't know why I put that in there again. Here's our first step. So step number 1. So I'm giving this to you in outline form. Make sure you put this in essay form if you're going to be asked for this on the test. So step number 1 is acetylcholine release. So as our action potential is going to reach the synaptic terminal of our motor neuron, again, that synaptic terminal being the distal end of the motor neuron that stores acetylcholine, it's actually going to cause calcium ions to enter into the cell. Okay, so the action potential actually causes the calcium ions to go into our synaptic terminal. The influx of positively charged calcium ions is what's going to stimulate the synaptic terminal to release the acetylcholine from the vesicles using exocytosis. And hopefully you remember exocytosis is the process for a cell to expend a little ATP, burn a little ATP in order to release something from the cell. That's step 1. Step 2 is activation of those acetylcholine receptors. So you can't see it in the picture here, but calcium was released into the synaptic terminal when the action potential reached here. That caused these vesicles to fuse with the synaptic terminal. They break open and acetylcholine is going to be released. Step 2, the acetylcholine is then going to diffuse across the synaptic cleft and bind to our receptors here. Okay, again, the synaptic cleft being the gap in between the motor neuron and the muscle fiber. When acetylcholine binds to the receptors, that's going to cause these chemically gated sodium ion channels to open. And again, so the motor end plate is the part of the sarcolemma with the acetylcholine receptors. I'm being very deliberate in my definition here because this is the stuff I want to make sure you put in your essays. Step number 3, once we've got our acetylcholine plugged into our acetylcholine receptors, then we can start our action potential in the muscle fiber. So when the acetylcholine plugs into these chemically gated sodium ion channels, the sodium ion channels are going to open up. Sodium is going to rush into the muscle fiber and depolarize just the membrane of the motor end plate. Okay, and that's good, but we're going to see we're going to want more than just our motor end plate to be depolarized too. So this then is going to cause rapid depolarization down our membrane, which is step number 2 of an action potential. Do you remember that? If you're having trouble remembering the steps in an action potential, please make sure you go back and look in the PowerPoints or look in the videos for this stuff because it is really important. That's why we talked about this previously, to make understanding what's going on in the muscle a lot easier. Okay, so once we've depolarized a small part of the muscle fiber, our motor end plate, we've got depolarization. We can now depolarize the next part of the membrane using voltage-gated sodium ion channels. So there's where our wave goes in. Remember, we'd used the wave as the analogy. So depolarize one part of the membrane, and then voltage-gated sodium ion channels, and then it depolarizes. And then voltage-gated sodium ion channels open, and then it depolarizes. And then voltage-gated sodium ion channels open, and it depolarizes. And we've got a wave of depolarization traveling down our membrane and going into the T-tubules. Remember we talked about those T-tubules in the first video? They're going to bring action potentials deep into the muscle fiber. Okay, and it's important that we include that the action potentials are going into the T-tubules. Step number 4, we need to stop the activity of acetylcholine, right? Because as long as acetylcholine is plugged into those receptors, we've got our voltage-gated sodium ion channels opening, sodium's rushing into the cell, it's going to stay depolarized. We can't repolarize it. So we've got to stop the activity of acetylcholine. And one of the ways that we do that is by using an enzyme. Now, I know this is a great big long word. It's called acetylcholinesterase. Okay, and the abbreviation is capital A, capital C, lowercase h, and capital E. Okay, how do we know this is an enzyme? Well, if you look at the last 3 letters of this word, ASE, anything that ends with ASE is an enzyme. So this is an enzyme that works on the substrate, acetylcholine. Okay, and by breaking down acetylcholine into its smaller parts, it's going to be released from the receptor closes, and then repolarization can occur. Okay, so the activity of acetylcholine is going to allow the membrane to be repolarized in case we need to depolarize it again. Is it bad if muscles contracted but did not relax? Well, a sustained muscle contraction without relaxation is called tetanus. Does that sound familiar at all? Well, perhaps you've heard of the disease tetanus, which is caused by a bacterium called Clostridium tetani. And I'm sure you've heard, you want to be careful not to step on old rusty nails or whatever, because if those materials have this fairly common soil bacterium, Clostridium tetani, on it, stepping on the nail is actually injecting these bacteria into your body, where those bacteria can begin to grow. And as those bacteria grow, they release a toxin, and this toxin causes sustained muscle contractions. So that's what the bacteria looks like under a microscope and with fluorescence there, so it shows up real well. And if you've got these bacteria growing in your body releasing their toxins, they're going to cause your muscles to contract and not relax. Is that a bad thing? You better believe it is. So here's a painting that was done by a British surgeon in the early 1800s. This was a British soldier who was injured, and from what we're told, it was a relatively mild injury. He was stabbed by a spear. It wasn't a life-threatening injury, but some tetanus bacteria were introduced into the wound through the spear. As these bacteria began to grow, they released toxins, and these toxins caused some really serious muscle contractions in this poor British soldier. The soldier ended up dying from tetanus, but as you can see, before he died, he had such strong muscular contractions on his posterior side. The only thing touching the mattress here are his heels, his elbows, his shoulders, and his head. Now, before he died of respiratory arrest, meaning his breathing muscles contracted and didn't relax, he actually fractured vertebrae in his back from those muscular contractions being so strong. Not a good thing, and certainly not a good way to go. But fortunately, we live in the 21st century now, so people don't really die of tetanus anymore, right? Unfortunately, that's not the case. Here is a picture taken by a professor from Emory University showing a man who is dying of tetanus. Here's a slightly older picture taken from the Centers for Disease Control website. This person is also in pretty late stage tetanus. You can treat tetanus if you catch it early enough with antibiotics, but the best way to prevent yourself from getting tetanus is to get the Tdap vaccine. Get the tetanus vaccine. It also includes a couple of other protections, so diphtheria and pertussis or whooping cough. So the public service message for the day today is make sure you get your vaccinations. You don't want to die from tetanus. People still do sometimes. All right, so let's look at the big picture here. Major steps in the contraction of a skeletal muscle. So we have stimulation of a muscle fiber by a motor neuron. And again, be able to talk about this as an essay question. We just finished this part, so let's change that to a check mark. Okay. I can tell you from when I learned this at the community college I went to way back in the 1980s, there's an awful lot going on here. So if you're not picking this up right away, don't worry about it. Go back through the notes, go back through this video, look in your textbook. There are other resources that may be available online that I may share with you in class. Make sure you get this part before moving on to the next step. Because if you don't understand this, it might be a little bit more difficult to understand what's coming up next. So I'll pause for a moment to allow you to stop the video if need be. Good. So let's move on to the second major step in the contraction of a skeletal muscle, and that is excitation-contraction coupling using the sliding filament model. And again, be prepared to identify and explain the steps in excitation-contraction coupling using the sliding filament model. All right, now some of this stuff should look kind of familiar from the first video. So here we see our myosin, our thick myofilament with the myosin head sticking up. Over here we see an actin, or a thin myofilament. It's made up of two twisted strands that are together called an F-actin or fibrous actin, and each fibrous actin is made up of individual beads here called a G-actin. We've got this long orange, in this case, rope-like tropomyosin that would be covering up any of the active sites or binding sites on the G-actins. And we've got this little structure right here, which is our troponin, that's going to help hold the troponin, or the tropomyosin and the G-actins together. So again, we got to know all this stuff in order to understand what's going on. If you're a little fuzzy on this stuff or want a little practice, I'm going to give you a moment to pause the video and refresh your memory before moving on. Okay, welcome back. So when we talk about excitation-contraction coupling, we're talking about the relationship between the delivery of an action potential from a motor neuron to a muscle fiber, and then what's happening in our muscle fiber in order for the sarcomere to contract. And we know that it contracts using actin and myosin sliding over one another, which we call the sliding filament model. And this is going to depend upon having calcium ions available to initiate this whole process. So calcium is going to be part of our switching mechanism to just get the muscles to contract when we want them to. Because if the muscles stay contracted all the time, we call that tetanus, and we say that's bad. Right? So we have to have a mechanism for turning on the contraction and then turning it off when we're done. So, first step in excitation-contraction coupling is the action potential moves along the sarcolemma and into the T-tubules. And if you've been paying attention, you'll notice that this is also one of the steps in stimulation of a muscle fiber by a motor neuron. Right? So I intentionally have this overlap here so you see the relationship between the two. So when we're stimulating a muscle fiber, we're going to see the action potential traveling down the sarcolemma, or cell membrane, and then down into the T-tubules. Okay? That action is essential in order to get the muscle fiber itself to contract. So that's why we see that again here. So the action potential moves along the sarcolemma and into the T-tubules, and it's going to be delivering its action potential into the muscle fiber there. When that happens, it's going to cause calcium ions to be released from the sarcoplasmic reticulum. So here again is our sarcolemma with our T-tubules. When an action potential travels along our sarcolemma here, the action potential is going to go down into the T-tubule. And if you remember, on either side of our T-tubule, we've got sarcoplasmic reticulum. And in fact, the relationship between these three structures together, T-tubule in the middle, sarcoplasmic reticulum on either side, we called that the triad. When the action potential travels down the T-tubules, it's going to cause the sarcoplasmic reticulum next door to release calcium. Step number 2. Those calcium ions are going to bind to the troponin in the thin filaments. And that's going to cause, so this is crucial, when calcium binds to troponin, that troponin is going to change shape a little bit. And by changing shape, it's going to cause the troponin-tropomyosin complex to shift a little bit. And when that troponin-tropomyosin complex shifts, it's going to be exposing the active sites on the G-actins. So here we see our myosin and the myosin head. We're not ready for this yet, but it's coming soon. Here we see our troponin, and these little dots right here represent calcium ions. When calcium ions bind to troponin, the troponin just changes shape a little bit. And by changing shape just a little bit, you've got all these binding sites on our G-actins are now exposed. So in a sense, the action potential is going to cause the release of calcium. The release of calcium is going to bind to troponin, and that's by changing the shape of the troponin, allowing our active sites or our binding sites on the G-actins to be exposed. So what's going to happen next is our myosin heads are going to be able to plug in, and using a ratchet-like motion, they're actually going to cause the actin and the myosin to slide over one another, at least a little bit. Okay, so first thing we're going to talk about is the cross bridge formation. So a cross bridge is what we call it when the myosin head here plugs into the active site on our G-actin. Okay, it's just the connection between the two. We call that a cross bridge. Once the cross bridge has been made, then we're going to have the power stroke. And you can't just call it wimpy power stroke. You got to put a little oomph behind it, right? So this is the power stroke. And the power stroke is where energy that has been stored in our myosin head is going to be released. And when that energy is released, it's going to pivot a little bit. It's going to shift. And by pivoting, it's going to cause the thin filament to be slid over the thick just a little bit. So here we see a cross bridge has been formed between the myosin head and binding on the G-actin. So in the power stroke, the energy that was stored, so ADP plus phosphate is ATP. Remember that molecule that we use for energy? Okay. When the energy has been stored in our myosin head here, when the cross bridge occurs, this energy is going to be released. So we're releasing the phosphate and the ADP here. That myosin head then can shift, moving the actin over the myosin a little bit. After we've had our power stroke, we now have what's called the recovery stroke. And it's in the recovery stroke that the cross bridge is going to be released. And careful, looky here. It's here that ATP is going to bind to our myosin head. And by binding ATP to our myosin head, it's going to bring the myosin head back to its original, sometimes we say cocked position, right? So we're storing energy in that myosin head so that if there's another binding site available, you can have cross bridge, power stroke, and then another recovery stroke. Okay, so the recovery stroke is where ATP is going to plug into the mechanism here and allow for other power strokes to occur in the future. So after the recovery stroke, our myosin head has had ATP bound to it. It's been moved away. It's now ready to make more cross bridges with a different part of the actin myofilament. And if this can happen over and over and over again, we're going to make our sarcomere shorter. Okay, so here you can see the cross bridge has been released after ATP binds to it. So here is our recovery stroke. And the myosin head, as it breaks down the ATP. So ATP is going to be released. Well, it's going to be broken down, but it will still be bound to the myosin head until a new cross bridge can be made. So step 6 here is that new power strokes are going to continue to slide the thin filaments over the thick again and again and again. And a really nice analogy for this is like you have hands in people that are playing tug of war. They're going to continue to make cross bridge, power stroke, recovery stroke, cross bridge, power stroke, recovery stroke again and again and again, like these folks are doing here, until acetylcholine esterase stops the mechanism here. So stops our action potential down the muscle fiber. It's going to stop calcium and allow for repolarization to occur. Okay, so we talked about step number 1, stimulation of a muscle fiber by a motor neuron. We just talked here about excitation-contraction coupling using the sliding filament model. So we'll put a check mark there. I can't say this strongly enough. Each one of these two parts, stimulation of a muscle fiber by a motor neuron and excitation-contraction coupling using the sliding filament model, each one of these is a potential essay question. So please practice putting each one in essay form. So I'm going to give you a chance to stop the video and go back and review if you like. Otherwise, if you're feeling good, we're going to talk about the next topic, which is muscle relaxation. So in order for a muscle to relax, you got to get rid of the calcium. So calcium pump, which is a form of active transport, is going to take and pump calcium from the neuromuscular junction back into the sarcoplasmic reticulum of our muscle fiber. And if the calcium is gone, calcium isn't bound to troponin anymore, so the troponin-dopamycin complex goes back to its original shape. And tropomyosin will then cover up the active sites on our G-actins, and no more cross bridges can occur if the binding sites are covered. And as a result, this prevents more cross bridges from forming. Have you ever heard of rigor mortis before? Rigor mortis is what happens to a body in the hours after death occurs. And it all goes to the fact that you're not doing aerobic respiration anymore. So you're not taking in oxygen, you're not breaking down glucose to make ATP. No more breathing, no more activity of the heart, no gas exchange. The oxygen in the body and in the muscles is going to be used up. Once there's no more ATP, you can't do a recovery stroke. So those myosin heads stay plugged into the last cross bridge that they made. You had a power stroke, but there's no ATP for the recovery stroke. So what ends up happening, depending on temperature and conditions in the hours after death occurs, those muscles start to lock up. And that's all due to the fact that you're not making ATP in order to do new recovery strokes. After a period of time, generally 24 to 48 hours, the muscles do relax as bacteria begin to break down those structures in the sarcomere. But the reason for rigor mortis all goes back to you can't do the recovery stroke. Like that right there. No recovery stroke can happen if you don't have ATP in order to break it and bring the myosin head back to its original position. One of the things we can do is we can measure what's happening in a muscle when it contracts. And this you can do in a laboratory. And I remember I did this as an undergraduate. We would take the gastrocnemius muscle or the calf muscle from a rabbit. We would submerge this muscle in an isotonic glucose solution. We would bring a wire, so a computer would actually generate a small electrical charge which simulates an action potential. And then we'd attach each end of the muscle fiber to a spring-loaded device that could measure muscle contractions. And when you would stimulate the gastrocnemius muscle of your rabbit, the muscle would contract and that would be measured. The graph that results from an experiment like this is called a myogram. And if you look on our x-axis here, this represents time in milliseconds, and a millisecond is 1,000th of a second. So we're talking really short periods of time here. And on our y-axis here, this represents tension on the muscle. With the higher up you go, the more tension, so the more the muscle is contracting. So a myogram is going to record the electrical activity of a muscle contraction. And as you can see from the diagram, there's 3 parts of a myogram. We've got the latent period, the contraction period, and the relaxation period. So the latent period, which you can see here kind of in a light yellow, is a very short period of time, maybe around 5 milliseconds or so. And this is the period of time between when the muscle is stimulated and when it actually begins to start contracting. So latent means, well, it's been stimulated, but it's not contracting yet. The contraction period, which goes on for another almost 20 milliseconds or so, is the period of time when the muscle is contracting more and more and more and more. And this is after one single stimulus. So it's simulating one action potential. After this, the muscle is relaxing and being stretched out to its normal length. And this can take, another 75 milliseconds or so to accomplish. So we're going to use this myogram when we talk about how in a laboratory we can do multiple things. We can send stimuli to a muscle and see what happens. So a twitch is what occurs. And again, this is happening in a lab. This would be hard to measure in a living organism. But because we can use computer-controlled stimuli in a muscle in the laboratory, we can see that a twitch is what you get after a single action potential. So it would be a single contraction. If you have a stimulus and the muscle begins to contract, and then another stimulus and the muscle contracts, and then another stimulus and the muscle contracts, and you do this so another stimulus occurs before relaxation can be completed, what ends up happening is the muscle contraction gets stronger and stronger and stronger. So this is going to cause increased tension, or sum. Remember, sum is adding. That's why it's called wave summation, because the muscle is not allowed to stretch back to its original length before another stimulus comes along. So here we can see, it's not exactly the same as the myogram we saw on the last slide, but you get the general idea. Each one of these arrows represents where another stimulus is applied. So you're applying a stimulus here, and you get a contraction. And then you apply another stimulus here before the muscle fiber completely relaxes, and you get another you get another contraction that's actually stronger. There's more tension in it. And another one's going to happen here, and then another one's going to happen here. So every time you do that, the muscle is going to contract with more tension or more force. And this process of sending these action potentials so fast that the muscle isn't allowed to relax is wave summation. Now, we can have incomplete or unfused tetanus, where we have a short relaxation time between multiple stimuli. However, if we have action potential after action potential after action potential after action potential, one after another, without time for relaxation, you end up with what's called complete or fused tetanus. And here's an example of complete tetanus. So you're getting multiple stimuli going towards the muscle. It keeps getting contraction with more and more and more force. But notice there reaches a point at which you can't contract any more forcibly. So muscles are only able to contract with a certain amount of force. Even when you're sending action potentials faster and faster and faster, there reaches a point at which you can't contract with more force. And you see on your myogram, you get a graph like this called complete tetanus. Okay, we're winding down here, folks, only a little bit more, so stay with me. Let's talk a little bit about fine motor control. So if you recall when we talked about action potentials, we said generally action potentials either happen or they don't. Okay, we called that the all or nothing principle. Well, the same thing is going to happen with muscles. You can't have a muscle fiber contracting weakly. Okay, muscle fibers contract either with all their force or they don't contract at all. But how do we differentiate between picking up something light, like a piece of paper or a feather, and picking up some great big hand weight? Right? So do we pick up the feather the same way we pick up the hand weight? Okay, if we picked up the feather the same way we pick up the hand weight, we'd contract with such force, we'd probably hit ourselves in the face. So we have to have the ability to differentiate between picking up something light and picking up something heavy. And it's not in the contraction strength of the muscle, because we know muscle fibers can contract all or nothing. But what we see happening, if we're picking up something light, we might only use a very small percentage of our muscle fibers. So my biceps brachii, if I was going to pick up a feather, I might only use about 2% of all my muscle fibers to cause the flexion, to cause this movement here. But in those 2% of those muscle fibers that I stimulate, they contract as strong as possible. On the other hand, if I'm picking up a great big 20-pound hand weight, okay, I might be using 80% of all my muscle fibers. And all 80% that are being stimulated are contracting as strong as they can. So a light object only needs to stimulate a few muscle fibers. A heavy object needs us to stimulate a lot of muscle fibers. And the ability to regulate how many muscle fibers we're going to need for a particular action is called recruitment, sometimes also called multiple motor unit summation. So recruitment is controlling the number of muscle fibers that we're going to stimulate. And again, picking up something that's heavy is going to require that we recruit a lot of muscle fibers. Picking up something light means we only have to recruit a smaller percentage. But maximal stimulus is the strongest stimulus that is going to produce an increased contraction force. And in the case of, let's say I'm at the gym and I'm pumping some serious iron here. If I'm picking up a heavy weight and I'm trying to do some biceps curls, I will need a maximal stimulus in order to get flexion in my arms at the elbow, because I'll need to recruit all the muscle fibers in my biceps brachii there to do that. That's not really me though. So what happens when we exercise, right? So we like to go to the gym and we like to get all big and buff, right? So contrary to popular belief, when you are exercising, building up muscle, you don't build more muscle fibers. It might be building a few extra, but generally you're not going to cause more muscle fibers to be constructed. Instead, if you're going and you're working out and you're going for the burn, right? You want to get all big and buff. What you're actually doing is you're ending up tearing some of your muscle fibers. Now, homeostasis demands that we maintain, our constant levels. So we don't like it when we damage some of our muscle fibers. So if you're exercising a lot, you actually start building more myofibrils in your muscle fibers. So the number of muscle fibers doesn't significantly change. But if you start packing more myofibrils in a muscle fiber, the muscle fibers are going to get bigger and have a greater ability to contract. And if you have bigger muscle fibers, what do you get? You get somebody you're not going to mess with. You got great big muscles there. All right, and a couple of other things you might want to look at for test number 2. Look at how the presence of an acetylcholinesterase inhibitor, so a chemical that would block the action of acetylcholinesterase, how that might influence what's going on at the neuromuscular junction. You might look at what happens to molecules that bind to and block acetylcholine receptors, and might want to look at what happens if you lose or destroy acetylcholine receptors, how that can influence what happens in muscle contractions. And that's all I have for you. I know muscles was a pretty big topic. You're going to need extra time to take a look at this stuff. Please don't hesitate to ask questions. Let me know if there's something I can do to help. Take care, stay healthy, and we'll see you around.